Sampling is a method of selecting subjects for a research study that involves choosing only a portion of the population we are interested in. Sampling allows researchers to make conclusions or inferences about a population without having to investigate everyone. Why not talk to everyone, you ask? Well, this process, called a census, is more costly and takes more time than taking just a sample. If done properly, sampling can also give us a representative picture of what's going on for the whole population without asking everyone. So sampling is often a preferred approach among epidemiologists. Defining your population of interest is the first step to effective sampling. Suppose we wanted to conduct a study to investigate the prevalence of smoking among Canadians who had a heart attack in the past 12 months. Our target population, the population to which we will try to generalize our results to, is Canadians who had a recent heart attack. Since sampling from across the entire country would be difficult, we might choose to focus on sampling only from hospitals in Ontario and Quebec. So our source population would be recent hospital patients across Ontario and Quebec. We might ask a random sample of hospitals in these provinces for their records in order to make a list of all individuals that met our criteria. This list is our sampling frame, and each individual in our sampling frame is a sampling unit. Finally, the specific group of people or sampling units chosen to participate in this study would be called our study population. When sampling, it is crucial that we consider internal and external validity. Internal validity asks, how well do the study results relate to the source population? So, in our hypothetical study, if the conclusions we draw from our study sample are representative of the other members of our source population, then our study would have strong internal validity. External validity asks, how well can the study results be extrapolated to the target population? So, in our study, if the conclusions we draw from our study sample are valid for members of our target population, then our study would have strong external validity. Now, there are two main forms of sampling: probability and non-probability sampling. The main difference between these two is that probability sampling involves some form of random selection, where each member of the population has an equal chance of being selected, while non-probability doesn't. Examples of non-probability sampling include convenience sampling, where participants are chosen because they're easy to get, like animals in a trap or herds close to a research facility, and purposive sampling, where specific participants are chosen because they have particular characteristics, such as when we are studying a rare exposure or outcome. This is because there are few of these individuals in a population, so we must deliberately select them, as a random process likely wouldn't produce enough people with the characteristics of interest. The important thing to remember with non-probabilistic methods is that all members in the population do not have an equal chance of being selected, and while these methods can be cheap and relatively easy to do, the results obtained may have poor internal and external validity, because those selected may be different from those who are not. Probability sampling addresses these limitations by employing the use of a random process. This helps ensure that any sample taken from the population will be representative of the population under investigation. There are simple, systematic, and stratified forms of random sampling. Simple random sampling is a process where all members of the population have an equal chance of being selected. One way this can be done is to assign a unique number to every member of the source population, and then randomly select one member at a time by matching a randomly generated number to the member with that assigned number. This is done until the number of desired subjects is sampled. Systematic random sampling involves the use of a sampling interval, where the first member of a population is randomly selected, and then the following members are chosen at regular intervals. For example, large animal veterinarians might use this approach to sample cattle and check for hoof problems as they enter into a new pen. They might start by choosing the second cow that enters, and then checking every third cow from then on. Stratified random sampling involves first dividing the sampling frame into subgroups or strata based on a factor like age or gender. It's used when we might reasonably expect the measurement of interest to vary between the different subgroups. Once separated, the study sample is then obtained by taking samples using simple or systematic random sampling from each stratum. Here, the percentage sampled within each stratum does not have to be the same. There are a number of different ways to sample from populations, and determining the best approach to use isn't always easy. Overall, what you know about your population. the resources available to you, an approach that will allow you to effectively answer your research questions, and impact on internal and external validity are all factors that you should consider when making your sampling plan.